All right, so good morning. So now what we are going to do is take a look at mutations in a little bit more detail. So at this point you've looked at this idea of transcription and translation, and you should have a good handle of what's happening there and the idea that we're taking DNA, making the RNA, and then that RNA is used to make the proteins. And then what we looked at on Monday, Tuesday was the regulation. So you hopefully understand the idea that not all proteins are made all the time, so there's not transcription happening all the time for every gene. It only transcribes the genes that are needed for the type of cell that it is. So for example, your beta cell made insulin, but the intestinal cell did not make insulin. Okay. Same thing with like hemoglobin. The red blood cell made hemoglobin, but the other cells did not. The intestinal cell, the smooth muscle cell, and the beta cell did not make hemoglobin because hemoglobin was just needed in the red blood cell. So what we're going to look at today is some specific mutations that lead to differences in organisms, okay? And then what we'll look at when we're in class together is to look at that globin protein a little bit more, the hemoglobin that's in red blood cells, and look at what happens when there's a mutation in that hemoglobin specifically. Okay, so that's what's happening in class, and then looking at the disorder of sickle cell anemia that happens when there's a mutation in that globin protein. So let's take a look at the assignment here. What you're going to do is you're going to use this Learn Genetics website where we've seen these differences in different proteins. So when we did this activity with the simulation, you saw that you could make mutations in the protein, and depending on the way that that depended on how that protein looked. Okay, so this is an example of one protein, and it takes on a shape. If I do a different random mix, I can change the shape again. Okay, and then I can change it a little bit more. So this one's getting a little bit more stringy. And this is just looking at whether they like water, which are the green ones, or they don't like water, which are the yellow ones. I could make it all hydrophobic, which means they don't like water, or I could do it this hydrophilic, okay, where they do like water and they start to stretch out. And if you watch, as I click on it, you'll see that the names inside, the letters inside, change. Okay, and so it changes the shape depending on which amino acids are there. So as that happens, then you have to remember that the order of these amino acids is dictated by the order of the DNA. So when you looked at the mutations on the mutations worksheet, either Monday or Wednesday, depending on when you did it, you saw that you could change a letter in the DNA, and that change in that one letter would lead to a change in the amino acid that it coded for. And so if we change the amino acid, it changes the shape of the protein, and the shape of the protein is what's doing its job. So what you're going to do is use this Learn Genetics website, and you're going to fill out this table. So you're going to look at specific proteins, and you do not need to copy down everything from the website. You're just summarizing it to help yourself understand a way to answer this big question. And the question is just, how do mutations alter the protein structure and then the function of the proteins? Okay, so you're looking at these different organisms. So when you click on it, it'll take you to all the different organisms you're going to look at. So when you open up each one, you've got the information here. So you might want to do this in a split screen, and then you can see. So like for the Shar-Pei dogs, this is the protein. Okay, it's this HS2. So you don't have to write out all of this information. You can just say it's the HAS2, okay, which is an enzyme in the skin cells. So describing just kind of the job of what that protein does. Then you've got the DNA, and so it tells you that it gets turned on, all right, by how much of that protein is, how long it's activated. So just like we saw in our paper clips, where sometimes it can be activated longer, or sometimes it can be activated slower, that this has a switch to turn it on. So you're going to summarize a little bit about the switch, and the idea that what happens is that if it makes too much of the enzyme, so it has too much of the protein, HA, then the skin gets really thick, and then that's when you get all the extra folds. So these Shar-Pei dogs that have all this folded skin are making a lot of the HA, which means that the gene gets turned on for a long period of time. Here's some examples in other species, so you can summarize that. And then in terms of this, if it's a variation or a disease or both. So it can sometimes lead to things like skin inflammation, but it's more just a variation in the structure of the skin. Okay, then you can click the X, go back, and you'll do the same thing here with the double-muscled cattle, extra-toed cats, and all the rest of these. All right, so you'll fill in your table summarizing what you're seeing with those traits. Once you have that, you'll then write an explanation. Okay, so for this, what you're thinking about is how do these mutations alter the structure and the function. So in all of those examples that you saw, what evidence did you see of proteins being changed from the sequence and also then their job? Okay, so you answer that and think about the gene regulation piece that we talked about with the paper clips going on and off of genes. You can talk about translation changes, the silent mutations, missense mutations, those kinds of things. Thinking about what kinds of mutations you've seen with each of those things happening, okay? And then what you're going to do is draw a model or create some sort of a diagram to show you this whole process. So the idea of going from DNA to a trait and what's happening along the way there. So thinking about the DNA to the transcription, the translation, the protein structure, and then how maybe it could then be altered, okay? Like the one that you did and looked at up here. So I hope that that's helpful to you, and I will see you the next time in class.